morning everybody. Today we are going to be discussing about the inhibition of enzymes. Enzymes are biological catalysts synthesized in living cells, and they are colloidal and thermolabile in action and which may be specific in their action. Okay. So enzymes are biological catalysts which is synthesized in the living cells, and they are colloidal and thermolabile and specific in their action. Normally, there are two types of enzymes based on the location of this enzyme. One is the intracellular enzyme, another is extracellular enzyme. Intracellular enzymes are the enzymes which functional within the cells where they are synthesized, whereas the extracellular enzymes are these enzymes which are active outside the cell. We consider all the digestive enzymes are extracellular enzyme with reference to all the proteolytic enzyme with example pepsin, renin, carboxypeptidase, chymotrypsin, etc. The nature of the enzymes and the properties includes the holoenzyme, apoenzyme and coenzyme. Holoenzymes are which made up of the functional part of the or functional unit of the enzyme, and apoenzyme is considered to be the protein part of the enzyme, and coenzymes are non-protein organic part or non-protein prosthetic group. So you see the equation is like this: holoenzyme which is divided into apoenzymes and coenzymes. And when you consider the active sites of the active site, or it is called as active center of an enzyme, which represents a small region at which the substrate binds and participates the catalytic reaction. That is, the active site is a small region of the enzyme in which the substrate binds. So it is otherwise called as substrate binding site. The features of enzyme substrate active site, the existence of active site is due to the 3D structure of protein resulting the 3D native conformation. The active site is made up of amino acid known as catalytic residues. Active sites are regarded as the clipped or crevices or pockets occupying small region in a big enzyme molecule. Active site is not a rigid in structure and shape. It is flexible to promote the specific substrate binding. Active site possesses the substrate binding site and a catalytically active site. So as you shown in this figure, the figure illustrates clear that the enzyme is a larger in size when compared to the substrate, and this substrate is trying to bind with the active site of the enzyme. That is, active site is otherwise called as catalytically active site. The active center can be divided into two sites. First, where the enzyme binds to the substrate, that is otherwise called as substrate binding site, and the second catalytic site which catalyzes the chemical reaction. The active site determines the specific and catalytic activity. Generally, the active site consists of 3 or 4 amino acid residues. They form temporary bonds with the substrate and residues that catalyze the reaction of the substrate binding site or catalytically active site. So enzyme inhibition is defined as the substrate which binds the enzyme to bring about the decrease in the catalytic activity of the enzyme. So they are organic or inorganic nature. The enzyme inhibition is broadly classified into 3 categories. One is reversible inhibition, second is irreversible inhibition, and third is allosteric inhibition. Why it is so called reversible inhibition? The inhibitor binds non-covalently with the enzyme, and the enzyme inhibition can be reversed if the inhibitor is removed. This reversible inhibition again be subdivided into 2. One is competitive inhibition, another is non-competitive inhibition. So when you go through the difference between competitive inhibition and non-competitive inhibition, in competitive inhibition the inhibitor which is closely resembles the real substrate is regarded as the substrate analog, whereas the non-competitive inhibition the inhibitor binds the active site other than the catalytically active site of the enzyme surface. The inhibitor competes with the substrate and binds the active site of the enzyme but does not undergo any catalytic reaction. the inhibitor has no structural resemblance with the substrate as far as non-competitive inhibition is concerned. So as shown in the figure, it is very clear that it is illustrating the competitive inhibition and non-competitive inhibition, how this inhibitor and substrate competing each other to binds with the catalytically active site of the enzyme. So that is based on the Km value. We can draw a figure or a graph based on the Michaelis-Menten equation. Michaelis-Menten equation is B equal to Vmax into substrate concentration divided by Km plus S. If you know these all the values which is plotted in the graph, we can find out the velocity of the reaction. As far as the Km values increases, the Vmax remains unchanged as far as competitive inhibition is concerned. If you consider the non-competitive inhibition, the Km value unchanged while the Vmax is lowered. It is very clearly illustrated in the figure. Okay, that means that is the active site is more, means competition is relatively less, so that it is so-called non-competitive inhibition. Whereas in the competitive inhibition, the active center is less and the competition is more. The examples for competitive inhibition, succinate dehydrogenase is the classical example for competitive inhibition with succinic acid as the substrate. Another is the malonic acid, glutaric acid and oxalic acid, which has this structural similarity with the succinic acid and compete with the substrate for binding at the active site of succinate dehydrogenase. If you see the structure of succinic acid and malonic acid, you can find out these are the structurally similar. Structural similarity is there as far as succinic acid and malonic acid is concerned. And another classical example is methanol toxicity. Methanol is found to be toxic in the body when it is converted to formaldehyde by the enzyme alcohol dehydrogenase. In this reaction, in this functional aspect, ethanol can compete with the methanol for alcohol dehydrogenase. Thus, ethanol can be used for the treatment of methanol poisoning. So, some of the antimetabolites what we are giving as an example for competitive inhibition. These antimetabolites are the chemical compounds that block the metabolic reactions by a inhibitory action on enzymes. So these are called as antimetabolites. These are the chemical compounds that block the metabolic reactions by their inhibitory action on enzymes. If you consider the non-competitive inhibition, there is heavy metal poisoning. Heavy metals are considered to be classical example for non-competitive inhibitors. Consider mercury, lead, arsenate. All these are the considered to be non-competitive inhibitors. Another class of inhibition is called as irreversible inhibition. The inhibitors binds covalently with the enzyme and inactivate them irreversibly. These inhibitors are usually toxic poisonous substance. Example, iodoacetase is an irreversible inhibitor of enzyme like papain and glyceraldehyde 3-phosphate dehydrogenase. Examples for irreversible inhibitions, you consider diisopropyl fluorophosphate is a nerve gas which is developed by the Germans during the Second World War. The diisopropyl fluorophosphate is irreversible binds with the enzyme containing serine at the active site. That example can consider serine proteases and acetylcholine esterase. So another class of inhibition what we call the suicide inhibition, which is a specialist form of irreversible inhibition in which the inhibitor is converted to more potent form by the same enzyme that ought to be inhibited. You can consider allopyranol is an example. Allopyranol is an inhibitor of xanthine oxidase to convert the alloxanthine, is a more effective inhibitor of this enzyme. Allopyranol is also being considered as the drug choice for gouty arthritis. So consider the allosteric inhibition here. You can see the two sites here. One is allosteric site, another is the catalytically active site. The inhibitor is binds compete to binds with the allosteric site, so that there is no action on the active site of the enzyme. So this is which is potentially different from the competitive inhibition. The allosteric inhibition, the inhibitor is interested to binds with the active site of the allosteric site, so that relatively that inhibition is less because there is no competition in the active site of the enzyme. And there is what we call the suicide inhibition. That is, in the suicide inhibition, normally this enzymatic reaction, whenever the reaction got over, the enzyme will be regained without undergoing any change. But in suicide inhibition, the inhibitor becomes the part of the product, so that it is called a suicide inhibition. That is all about the inhibition of the enzymes. Thank you very much.